introduce our speaker today.
Howdy. Hey, howdy.
Dr. Jatin Roper. Dr. Roper is a clinical gastroenterologist at the Tufts Medical Center. He's also the director of the Center of Hereditary Gastrointestinal Cancer. He's a research affiliate at the Koch Institute of MIT, as well as an assistant professor of medicine. His research interests include metabolic regulation of stem cells and colon cancer using a mouse model in colorectal cancer. He also has experience pertaining to colonization of H. pylori and ghrelin and leptin. The pathways related to colorectal cancer, tumorigenesis, and intestinal cancer in both obesity and calorie restriction. Today he'll be talking to us about his work on the dietary controls of stem cells in both cancer and obesity. Please welcome Dr. Jatin.
Thank you. Thank you, Anna, for the introduction. And of course, I'd like to thank Rob Chapkin for the kind introduction and invitation to speak today. I had the privilege of meeting him when he visited us in Boston, so I'm really happy to have the chance to return the favor. Today I'll be talking about our work on dietary regulation of stem cell function in cancer. And I think the big question that faces all of us in the nutrition field is obesity. As Rob reminds me, this is especially a health challenge here in the South, but it's also a problem throughout the United States and the world. And what all of us are aware of is that obesity is one of the largest modifiable risk factors for cancer after smoking. And as smoking rates go down, we really see diabetes and metabolic syndrome and obesity being the largest driver of preventable illness, in particular cancer. We focus on colorectal cancer as a model system. We see a relative risk about 1.5 to 2 in both men and women for obesity. So when we're thinking about cancer, the way we like to approach it is understanding the progression from the normal epithelium to cancer. And so this is a stepwise progression of oncogenesis, first defined by Bert Vogelstein and his colleagues now over 30 years ago. And what we understand is that the normal epithelium first develops into aberrant crypt foci and polyps through loss of the tumor suppressor APC, which I'll go into more detail about. These polyps then acquire mutations in oncogenic KRAS, and then larger polyps, more advanced polyps, then lose P53 function or acquire mutations in PIK3CA or other mutations. So by the time you've gotten to that a large cancer, you can't really prevent disease through colonoscopy. At that point, many of the cancers have metastasized. And so despite our best efforts through colonoscopy and cancer prevention, we're still left with 50,000 Americans a year dying of colorectal cancer. So it's very important for us to understand the mechanisms in the early stages of oncogenesis. That is, how does obesity and diet regulate the normal intestinal epithelium and the initiation of tumorigenesis through loss of APC. So what does APC do in the intestine? So this is a general slide talking about what APC does in the Wnt pathway, both in embryogenesis as well as in other parts of the body, including the intestine. So what we understand now is that in the setting of a wild-type APC or no Wnt ligand, that the beta-catenin protein is bound to what's called the destruction complex, composed of axin 2, GSK3 beta, and other proteins. And then beta-catenin is then phosphorylated and then targeted for proteasomal degradation. In the setting of active Wnt ligand at the LRP Frizzle receptor, or in the case of inactivating mutations in APC, what happens is that the destruction complex is sequestered, beta-catenin is then free to translocate into the nucleus, where it activates transcription of Wnt target genes. And this is going to be important for the rest of my talk. And this is essentially the basis of how polyps form in the intestine. And Wnt plays a role in many other cancers and developmental pathways. So the question that we'll be addressing today is, what is the role of diet in intestinal tumor initiation? And then when you think about this, I want you to go back to the slide, because the cancers are progressing from normal mucosa to adenomas through loss of the APC gene. So when we're thinking about tumor initiation, we need to start by understanding the normal biology of the intestinal epithelium. And much of this work was described by Hans Clevers and his colleagues in the Netherlands. And what we understand is that in the small intestine, we have invaginations called crypts. And the base of the crypt is composed of intestinal stem cells that are adjacent to their niche cells called Paneth cells. And so here the stem cell is a narrow cell in green, and then the Paneth cell contains granules, is located adjacent to it. What we understand is that these stem cells are the cells of origin for all the differentiated cell types in the crypt and in the villus, which is composed largely of absorptive cells. So when you drink water and eat food, most of your nutrients are absorbed by the villi in the small intestine. Those cell types include absorptive cells, goblet cells, secretory cells, as well as the niche Paneth cells. And the Clevers lab developed an ingenious mouse in which these stem cells are marked by the protein called LGR5, which itself is a Wnt target gene. And they marked these cells with GFP and also added a tamoxifen-inducible allele so that you can cross this mouse to other genes, flocks, genes of interest, in order to drive expression specifically in the stem cell compartment. And so for the purposes of my talk, the key points here in terms of the language I'll use is when we do FACS sorting of these crypts, we can identify the stem cells as having high GFP expression. The daughter cells from these stem cells, also known in the literature as translate amplifying cells or progenitor cells, can be identified as having low GFP expression. And then once these cells differentiate, they then lose their GFP expression. So we can do this. It's a very powerful mouse that allows us to both identify, sort, and analyze the stem cells at the base of the crypts. The colon is very similar to the small intestine, with the key exception that there are no villi in the colon. We only have crypts. And Paneth cells are not located in most of the colon, but there are other Paneth-like cells that play a key role in there. One last point on the slide is that the Paneth cells have a very important role in supporting the stem cells. They secrete JAG1, JAG2, and Wnt ligands, in addition to other proteins, in order to support the function of the stem cells. So the Paneth cells are very important for the function of the intestinal stem cells. So using FACS, we can then do experiments where we can sort out the GFP high stem cells as well as the Paneth cells. So we can use CD24 as a marker for the Paneth cells. We can then mix Paneth cells and stem cells in a three-dimensional matrigel culture and ask the question, what is the regenerative or self-renewal potential of these stem cells by identifying formation of organoids or mini-intestines. And so organoid cultures have become a widely used model of intestinal stem cell self-renewal capacity as a proxy for their function in vivo. And we can also grow these mini-intestines from the intestinal crypts directly. And so we asked the question, what role does a high-fat diet play in the function of the intestinal stem cell compartment? And what we found, which is very similar to findings from Rob Chapkin's lab, is that high-fat diet increased by almost 2/3 the numbers of intestinal stem cells. And so on the y-axis, you have the percent of intestinal crypts from these mice that formed organoids. The percent of the cells sorted that were GFP high, identifying the stem cells, and the percent of the cells that were GFP low, which identifies the daughter cells or the progenitor cells. So what we concluded is that high-fat diet increases numbers of both the stem cell compartment as well as the progenitor cell compartment here. And we did similar experiments in the colon and found very similar results. We next asked, if high-fat diet is increasing stem cell numbers, does it also have an effect on the organoid forming capacity of these cells? And so what we do is then, during the FACS analysis, we sort the GFP high stem cells as well as Paneth cells, and then do co-culture experiments. Now, when you grow stem cells alone without their Paneth cell niche cells, the organoid forming capacity is very low. And so in the first bar you see very, very rare organoid formation. When you mix stem cells with Paneth cells, you get about a 15 to 20% organoid formation. And that's significant and expected. What we found that was really interesting is that in the setting of a high-fat diet, these stem cells acquire the ability to form organoids independent from the panicell niche. And this is very exciting because it suggests that the diet is in fact rewiring the function of the stem cells so that they can grow independent of the Wnt and other signals that are produced by their niche cues. And as expected, when you add panicels to the high-fat diet stem cells, that also increases organoid forming capacity. So our diets are composed of many different substances, and high-fat diet that we give to mice is composed of many different lipids. And so we wanted to identify the components of the high-fat diet that are having this effect on stem cell function. Now, the diet that we give to the mice is essentially purified lard. I don't know how similar that is from a Texas diet, but it may be a little close. I guess that's not fair since I'm not from here. But essentially, the mice are getting pure lard without the barbecue sauce. And so what we did was we took the different components of lard, of the high-fat diet, and tested them in vitro. And so we exposed these organoids in culture to palmitic acid, which is the predominant component of the high-fat diet. And so what effect does this have on stem cell numbers and function? And in vitro, we saw an expansion of the percent of the GFP cells that were, percent of the organoid cells that were GFP positive, suggesting an expansion of stem cell numbers within the organoids, shown and also identified on FACS analysis. And then when we sorted these cells from the organoid cultures and then grew them as secondary cultures, we found a dramatic boost in organoid formation per stem cell in the organoids treated with palmitic acid. So this suggests that palmitic acid may be one important driver of the high-fat diet phenotype in the stem cell niche. We also found similar results from oleic acid, other lipids that we found in a lipid mixture. And we also did studies in organoids derived from human colonic crypts. Let's see. Oops. Oh, shoot. Adam here? Did you leave? Oh, here we go. Okay. So we then tried to identify a mechanism that would explain how these fats are affecting stem cell function. And so we performed RNA-seq analysis of sorted stem cells and progenitor cells from control and high-fat diet mice. And one thing that was striking was a robust activation of target genes of the PPAR-delta signaling pathway. So PPAR-delta is a nuclear receptor that interacts with retinoic acid, and it responds to increases in fatty acid, and different targets of the PPAR-delta signaling pathway are involved in fatty acid uptake and fatty acid beta oxidation as well. And you could hypothesize a link here because PPAR-delta is intimately involved in fatty acid oxidation. And to validate our results from the RNA-seq, which I'm not showing you here, we did Western blot and qPCR to show that high-fat diet induces PPAR-delta targets. These include CPT1A, HMG-CS2, and FABP1. And we saw this induction both in intestinal stem cells and in their immediate progenitor cells. So then the question is PPAR-delta necessary, sufficient, or both to recapitulate the effects of the high-fat diet in the intestinal stem cells? And so to answer this question, we used a PPAR-delta agonist. So we were fortunate that a reasonably specific agonist of the PPAR-delta pathway is available, called GW501516, that has very limited activity against other members of the PPAR family, such as PPAR gamma and others. I should also mention that PPAR-delta is a predominant member of the PPAR family that's expressed in the colon and small intestine. And so when we gave this agonist to mice, we recapitulated our findings in the high-fat diet setting in that GW-treated mice exhibited niche-independent organoid formation, meaning that sorted stem cells from PPAR-delta agonist treated mice were able to robustly induce organoid formation in vitro. And we examined this question as well in human organoids. And so these are organoids that are derived from crypts from patients undergoing colonoscopy. And so we treated these organoids with lipid mix, palmitic acid, or the PPAR-delta agonist, and we found similar results on the colonogenicity or organoid forming capacity of these organoids. So, is PPAR-delta necessary for this phenotype? Well, to answer this question, we made an intestinal specific knockout of PPAR-delta. So this is a PPAR-delta flox mouse that's crossed to a villain-Cre promoter. So villain is a protein that's specifically expressed in the intestinal epithelium so that we can mitigate or eliminate possible effects of this pathway in the rest of the body. And so what you can see here is that in the PPAR-delta intestinal knockout, that the effects of palmitic acid, lipid, and PPAR-delta agonist are essentially abrogated once you lose PPAR-delta. So thus concluding that PPAR-delta is necessary for the fatty acid mediated phenotype on intestinal stem cell renewal. So this is all very interesting, but we wanted to go back to the idea of the effect of high-fat diet on the progenitor cells. So as I mentioned before, the LGR5 high stem cells are the cells of origin of all the cells in the intestinal epithelium, and they're also the cells of origin of intestinal cancer. And so if you delete APC or activate Wnt signaling in the intestine, you get cancers because you're activating Wnt signaling in the intestinal stem cells. The differentiated cells in the intestine do not have the capacity under normal physiologic conditions to induce cancer formation. But what was interesting is that we found a dramatic increase in organoid forming capacity in the progenitor cells or daughter cells, which are normally not stem-like. And we found this effect both in the high-fat diet setting as well as with PPAR-delta agonist. Here we see a 4 to 6-fold increase in organoid forming capacity in the GFP-low progenitor cells. And so this raised the question of whether high-fat diet or PPAR-delta signaling is affecting the expression of beta-catenin target genes, which are involved in colon cancer initiation and progression. And this is single-cell analysis that we performed with Fluidigm for beta-catenin target genes. And this figure just is basically showing you that there's an obvious distinction from the control stem cells and progenitor cells and the high-fat diet treated stem cells and progenitor cells. When we look more closely using violin plots, and so here's again the single cell analysis. So on the y-axis is expression of a selected number of beta-catenin target genes, and then the width of the plot is the number of cells that have that expression. And so the fatter the plot, the higher up it goes, the greater expression in more of the cells analyzed. And so BMP4, JAG1, JAG2 are all beta-catenin target genes. And what you can see from this figure is that there is robust enhancement of beta-catenin target gene expression in the setting of both high-fat diet and in enforced PPAR-delta signaling in both stem cells and in their progenitor cells. And I can also add that JAG1 and JAG2 are also, in addition to being beta-catenin target genes, are also involved in Notch signaling and are important products of Paneth cells. I didn't show you here, but we also found that high-fat diet decreased numbers of Paneth cells. So what this suggests is that the stem cells are, as they become niche independent, they don't need the Paneth cells to support them anymore. And one way that they do that possibly is by producing JAG1, JAG2, and other important self-renewing ligands on their own. And this is just one example of how we validated this using histology. So we did in situ hybridization for JAG1, and so each dot is supposed to be one mRNA particle or a few particles. And here you see robust enhancement of JAG1 expression in the high-fat diet treated mice. And we saw similar results in mice that were treated with a PPAR-delta agonist. So this is a general paradigm that we can use to understand the connection between a high-fat diet, PPAR-delta, and tumor initiation. So in the normal setting, so here blue are non-stem cells, red stem cells, you need to hit the stem cells in order to initiate the tumor. Typically that would be through loss of APC or activation of Wnt signaling. In the setting of diet-induced obesity, so number one, you have an increase in the stem cell compartment. So you have more stem cells that are available to initiate cancer. And then number two, we hypothesize that the non-stem cells may acquire stem cell-like properties in order to initiate cancer. And so we next wanted to test this possible hypothesis for why high-fat diet and PPAR-delta signaling could increase tumor initiation in the intestine. We, as I mentioned to you earlier, obesity is a well-known risk factor for colon cancer initiation. There can be many, many reasons for that, immunologic, stromal, or it could be from obesity itself. We fed mice on a high-fat diet for up to 9 months, and what we found is that it dramatically increased spontaneous intestinal cancer formation, thus suggesting that high-fat diet not only affects stem cell dynamics, but also promotes tumor initiation. When we examined polyps from mice on a high-fat diet versus control, we found robust induction of PPAR-delta target genes. Here we're looking at in situ hybridization for CPT1A. And so this suggests a link between our findings in the normal intestine and possibly cancer. And so to answer this question, we set up the following experiment. We took APC flox mice, crossed them to the LGR5 GFP mouse, and the control group got vehicle treatment, and the treated mice got the PPAR-delta agonist. And we gave the mice 2 doses of tamoxifen in order to delete APC only in the intestinal stem cells. And so 4 days after that, the APC gene is lost, only in the stem cells. 4 days after excision, we then sorted the GFP high stem cells and the GFP low progenitor cells. We then transplanted these cells into recipient mice. And we asked the question, does enforced PPAR-delta signaling affect the ability of these cells to initiate tumors? And so you hypothesize that only the stem cells that lose APC would have the ability to initiate tumors, whereas progenitor cells typically do not, because they don't have stem cell initiating functions normally. But what we found was that in the setting of enforced PPAR-delta signaling, that these cells acquired stem-like properties in the setting of APC loss, and were able to initiate tumors at the same rate as the GFP high stem cells. And so together, what this work suggests is that high-fat diet and high-fat diet activated PPAR-delta enhances the stemness and the tumorigenicity of the intestinal progenitor population. And so what excited us about these findings is that we're showing for the first time, using the intestine as a model system, that number one, stem cells can gain niche independence in the setting of a high-fat diet and PPAR-delta signaling. And then secondly, that non-stem cells, that is progenitor cells, can acquire stem-like properties, not only for self-renewal, but also to initiate cancers in vivo. So I'll talk a little bit about another project that isn't published, that is related to the transplantation model that I described. So gene sequencing studies have identified many different genes that can be involved in tumorigenesis, tumor suppressors, and oncogenes. This is just one example from Genentech a few years ago. We've studied KRAS, P53, PIK3CA, and classic mouse models, but there's many, many other genes that are left to be studied, but have not been because of the limitations of the mouse models that we have. And so this is a little bit more detail about the transplantation models that we've developed. What we do is we are able to insert injection needle using mouse colonoscopy into the mucosa of the mouse, and then inject cells or virus in order to develop new tumor models. And so what this does is it induces a bubble formation that we can see during colonoscopy. And to show you how specific this approach is, we injected adenocre into luciferase reporter mice. And so what you can see up here are very discrete circles, which is the extent of recombination from the injection. So what we're showing here is that using the colonoscopy injection approach, we can induce site-directed recombination in the distal colon. And if we inject CRE, a lentiviral directed CRE recombinase, we see a recombination in tomato reporter mice at the base of the crypts, which is where the stem cells are located. Here's a Paneth cell and here's a stem cell. So if you can induce recombination in the stem cells, you would hypothesize that losing APC in those cells would induce tumor formation. And that's exactly what happens. About a couple weeks after mucosal injection of lenti-CRE, we see robust tumor initiation at the base of the crypt, in the distal colon of these mice. Now, I'm not showing the slides, but we have also done similar experiments by injecting adenocre into APC flox mice, and we can also deliver activated tamoxifen into mice in order to induce tumor formation. So these models are different ways of delivering CRE recombinase to APC flox mice to develop colon cancers that are single and are located only in the distal colon, where we can watch them with colonoscopy. This is in contrast to traditional models like the APC min mouse, where the mouse develops many, many polyps throughout the small intestine and takes 2 to 4 months to develop. We then decided to take this work one step further and ask the question, can we use CRISPR gene editing in order to induce tumors in mice that do not have cancer predisposing genes? So just a quick summary for the graduate students here about the CRISPR system. So there's 2 components. There's the Cas9 endonuclease, which is an enzyme derived from prokaryotes, of bacteria, to protect the bacteria against invading viruses, and it's been harnessed to edit genes in mammalian cells. And so the Cas9 protein is directed to the site of interest by the guide RNA, which is the 20 nucleotide sequence that's complementary to the site of interest, and that's adjacent to a 3 nucleotide PAM sequence. So basically, if you have your gene of interest in Cas9, you can edit any gene of interest. And so what we did was we made a lentivirus that has a guide against the APC tumor suppressor gene and also Cas9. And using that mucosal injection approach that I described to you, we inject the virus into the mucosa of wild-type mice, so they don't have any cancer predisposing genes, and we see tumor formation in the distal colon that's GFP positive. And when we look at the histology, we see an adenoma, or a precancerous lesion that shows nuclear beta-catenin localization, which is a hallmark of Wnt signaling and tumor formation that shows GFP positivity. There may be some silencing of the lentivirus, which explains the mosaic expression here. So to prove the CRISPR deletion, we performed sequencing of the site in the APC gene. And what's interesting about this is that we're able, because each cell has undergone CRISPR editing, it's essentially barcoded or tagged. And so by identifying the number of clones, we can identify the number of cells of origin in this polyp. And what we see is that in these polyps, there's only 2 clones of origin that initiate the entire tumor. Here's the sequencing results that shows insertions and deletions at the site of interest in the APC gene. Another approach in order to increase titers of the lentivirus is to deliver the guide APC by itself to a Cas9 expressing mouse. So this mouse was made by a colleague at MIT. And it's a lock-stop locks Cas9 GFP. So when CRE is expressed in the tissue of interest, it activates the Cas9 enzyme and is fluorescent with GFP. And so in this setting, we see robust and more efficient tumor formation that's GFP positive. Now, when we infect the epithelium with lentivirus, it's very promiscuous. It infects the stromal cells, epithelial cells, muscular cells, all those cells. And so we wanted to develop a way to limit CRISPR gene editing to the epithelial cells, which is where we want to delete APC to induce tumors. And so what we did was we crossed the Cas9 mouse with a Villain-Cree mouse. I mentioned earlier that Villain is an intestinal-specific reporter. And when we give this mouse tamoxifen, it then activates Cas9 only in the epithelium. And so the epithelial cells turn green. When we deliver a lentivirus that has turbo RFP, what we see is tumor formation. And then on fluorescence, what we see is infection of stromal cells, they're red. The epithelial cells here are green, expressing Cas9. And then yellow is the epithelial cells that have undergone CRISPR-mediated deletion of APC. When we sequence these tumors, we find up to 10 to 12 clones of origin due to the higher viral titers that we're able to deliver to the mucosa here. So having developed technologies to induce CRISPR editing with lentiviral delivery, we asked the natural question, which is, can we deliver organoids in order to develop an in vivo model of organoid function? And so we first took normal or wild-type organoids, and we transplanted them into immunodeficient mice, and then we sectioned the mice to see if we could get engraftment of these organoids. And here we see TD tomato positive organoids engrafting into the lamina propria of the recipient colon, right here. And these organoids are small intestinal in origin, which you can see the lysozyme is staining the Paneth cells in the organoid here. So having developed ways to transplant wild-type organoids, we then asked, well, can we develop a new model of colon cancer by deleting APC in organoids? And so we can use CRISPR in vitro to delete APC in organoids, to develop APC null organoids, which then grow without Wnt signals or our spondin, which is an activator of Wnt signaling. We then transplant these organoids into syngeneic recipients, and we see efficient and rapid tumor initiation in the recipient mouse. And so we think this is very exciting, not only as a model of colon cancer, but also a way to study diet and aging and other metabolic effects by separating the epithelial cells of the tumor from the stromal cells. So for example, one could transplant organoids from a mouse on a high-fat diet to a thin mouse, or vice versa. You could also do manipulations in the organoids in vitro, genetic or metabolic manipulations, and then transplant those to test their function in vivo. And so we're using this model for a variety of applications that weren't possible before using traditional genetically engineered models. We then wanted to apply this for more translational studies, potentially for therapeutic application with patient tumors. And so we've developed ways to derive organoids from cancers, from patients who are having surgery for their colon cancers. And we can then take these organoids from the patient, then transplant them into immunodeficient mice. So the traditional approach to model patient-derived cancers is to take the tumors, digest them, transplant them underneath the flank of a recipient mouse. While those grow tumors, we would argue that it's not a perfect model because you're not modeling the stromal environment of the colon. And so when we transplant these mucosally via colonoscopy, we see robust tumor formation in the distal colon. It's CDX2 positive, indicating intestinal origin. And we just did a human keratin stain to show the human origin. We then asked, well, if we're transplanting those into the colon, would these cancers invade and possibly even metastasize? And we are excited to find that once you observe these cancers for up to 2 months, that a subset of them invade into the local vasculature and metastasize to the liver. And then the histology of these liver tumors are essentially identical to the primary tumor that's located in the distal colon. And we observed this in about 20% of the mice that received the transplant, which is not high, but we think that reflects the biology of metastasis, where most cells are not competent to invade and then spread to the liver. And so we're now doing follow-up studies with larger cohorts of patients with different mutations, and to see whether that affects their ability to metastasize. So that's my talk. I'd like to thank our mentors and collaborators at MIT, the David Sabatini Lab, Mary Yilmaz, other members of the lab, and collaborators at Tufts and our clinical sites at Mass General and at Tufts Medical Center. Thank you very much.
Thank you. So this presentation now is open to question. Rob?
So let me start off. Fabulous presentation.
Thank you.
Very exciting data. I commend you on all the very novel approaches you're taking. So I was just wondering, if you take you generate an organoid from a high-fat animal, do you have any idea how long it maintains this advantage? Is this does this dissipate over time with passing of the organoid, or is this what you would say a permanent change in the steady state dynamics of that complex system?
Yeah, great. So Rob's asking whether the effect of the high-fat diet in the intestine is permanent, or if there is some perhaps epigenetic silencing that would limit its effect long term. So we're currently doing studies where we take mice off high-fat diet, and it appears to be that the PPAR-delta program is active for at least 4 to 6 weeks. Then after that, there may be some loss of it, but we're still working out the details of that. That's a great question, though.
Yeah, 2 questions. Very nice talk. Impressive. So usually high-fat diet promote cancer. So whether a high-fat diet in intestine can activate the beta-catenin pathway.
Right.
So whether the high-fat cell or fatty acid palmitic can activate the beta-catenin pathway. Number 2 question is, if you drive PPAR-delta, you think PPAR-delta is a mediator. So if you overexpress the PPAR-delta in epithelial cells with your vivian like a promoter region, whether overexpressing PPAR-delta in that kind of region can promote cancer.
So the first question was whether the diet can activate beta-catenin, right? Yeah. So I just showed a little bit of the data showing that both high-fat diet and the PPAR-delta agonist promote transcription of beta-catenin target genes. So what we did was we selected 48 beta-catenin target genes and then did single-cell analysis in about 200 cells and showed robust expression of beta-catenin target genes. We also did immunoprecipitation studies showing direct interaction between PPAR-delta and beta-catenin, thus supporting the hypothesis that PPAR-delta is activated by both high-fat diet and PPAR-delta agonist. What was the second question?
Second question, whether PPAR-delta activation is sufficient to cause cancer. So usually many data I think is necessary is because PPAR-delta activation involving hepatitis, fatty acid signaling pathway is well known. But whether this signaling pathway is sufficient to cause cancer.
Yes, that's a good question, right? So so in this study we show that enforced PPAR-delta signaling bestows tumor initiating capacity on the progenitor cells. And we're currently doing similar studies in the PPAR-delta knockout mouse to see whether PPAR-delta is necessary for the phenotype. So we're taking PPAR-delta knockout mice, putting them on a high-fat diet, and seeing whether the PPAR-delta is necessary for the tumor initiation capacity, or possibly there are many other pathways that high-fat diet activates that may promote tumorigenesis. Our work doesn't obviously obviate the effect of a diet on the immune system or on other signaling pathways, but it shows that PPAR-delta certainly may be an important mediator of the phenotype.
So another question is, because PPAR agonist like cyclitazone are well-known anti-diabetic drugs. So right now in the clinical trials or in the clinic,
The
So whether on the diabetic drugs like TZD, those kind of drugs can prevent the hyperglycemia, treat
Right.
diabetes, whether those kind of drugs can promote the cancer, particular kind of cancer in your case.
Yeah, for you mean the PPAR gamma agonist? Yeah. Yeah. Another good question.
Well, PPAR-delta, I think, so no.
Yeah.
PPAR alpha, gamma, delta, so they have different
Sure.
overlapping functions too.
Sure. Yeah. So the question is about the other PPARs, alpha and gamma. So their expression is lower in the intestine, and so which is why we hypothesize that PPAR-delta might be the most important pathway in the intestine. But we're actually treating mice with rosiglitazone to see what the effect is. So we don't have those results quite yet. I should mention that we also looked at the DB mouse, which is an obese mouse that's on normal chow. And so if you thought that the effect of the high-fat diet was only through obesity and not through the dietary components, then you might expect to see the same phenotype in the DB mouse. But in fact, we did not. We saw slightly decreased stem cell numbers in the DB mouse, and we didn't see the same effect on the progenitor cells. But those are limited studies, and we're doing more studies with the DB mouse.
Nancy?
Yeah, I'm just curious, the transplant studies that you're doing, is it like everything that you're transplanting is below the surface of the epithelium? Have you attempted to try to do the transplants of the organoids into the actual surface epithelium so that those cells really do have the full colonic environment?
Yeah, so I think the best we can do is to get the cells into the lamina propria, which is right underneath the crypts. And then what we observe is that if those cells are tumorigenic, those organoids, then they outcompete the native epithelium and they invade to the surface. When we transplant normal organoids, it seems like they mostly stay in the lamina propria. Yeah, but I don't think there's a way to deliver more superficially. Another approach, and others have done this, is to give mice colitis through DSS colitis, and then do an enema of a lot more organoids than we give, a lot of organoids, thousands, and through the colitis, the organoids can then stick to patches, and in that case, they integrate into the epithelium.
I'm sorry if I missed it.
Sure, no problem.
What was the approximately the time that you had the mice on high-fat diets?
Yes, the question is how long we had the mice on high-fat diet. I know I didn't mention that. Thanks for asking. So we looked at both short-term and long-term, and most of the effects were found with long-term high-fat diet, which is about 6 to 9 months. But some of the phenotype can be found with short-term of about 1 month. We did many of the follow-up experiments with the GW PPAR-delta agonist partly because those mice only needed about 4 weeks of the agonist, so we could complete the experiments more quickly. Yes.
I actually like to follow that question. So if that type of a feeding time period actually generate significant increase in inflammatory state, particularly pro-inflammatory cytokine expression at high level, have you ever get a chance to test?
Yeah.
So not just palmitic, but also the cytokine effects on those stem cell and also the progenitor cell.
Yeah. So
on that?
right. So the question is about the effects of the inflammatory environment on the stem cell compartment. To answer your question another way, we first asked whether the high-fat diet induces inflammatory response in the stem cell compartment. So we know that, of course, the high-fat diet does induce inflammation in the stroma around the stem cell compartment. When we looked at FACS-sorted stem cells and progenitor cells, we didn't see a difference in phospho-STAT3, NF-kappa B, I-kappa beta, and other inflammatory markers. And to answer your question, whether inflammatory cytokines affect stem cell function, it appears that these are other experiments that are not related to this, but it appears that IL-1 beta does activate stem cell renewal, but those are really preliminary experiments.
Thanks. Any other questions? Yes.
I just have a question, more technical question. You were talking about the APC blood plasmide
Yeah.
that you induced to like them creating this excess the null model, then you do the transplant. If I remember correctly, you said the day 4 that you
Yeah.
do the transplant.
Correct.
I understand that like the intestinal cells goes
Yeah.
And
So,
we can study that you, at that point, using all the cells become a null cells, or?
yeah, so the reason we did that is because we wanted to not have actual polyps to form, but we wanted to only lose APC in the stem cells and their immediate progenitor cells. So if we were to wait, let's say 2 weeks, right, then we would be sorting out all cancer cells, right? So we wanted to sort out stem cells and progenitor cells that have just lost APC function.
Do you have idea what's the percentage of cells that need to be sufficiently accessed?
Oh yeah, essentially, so once they've gotten 2 doses of tamoxifen, it'll delete APC in all of the all cells that are LGF5 positive. So all the cells that we sort will be APC null. Yeah.
Another question? Let's say again.
Yeah, so I'm curious on the studies where you were trying to test the effect of palmitic acid.
Sure.
You also had another treatment that just said lipids.
Yeah, that was like a lipid mixture. It has oleic acid, palmitic acid, and other acids.
Fatty
Yeah,
acid.
but the only specific ones we looked at were palmitic acid and oleic acid. Yeah. Yeah.
Any other question? If no, then we're done. Let's just thank the speaker for the outstanding presentation.
Thank you.
So graduate students are invited for the informal meeting with the speaker at table chair 124.
Oh, thanks.
Thank you.